All right, everybody. So I guess we're getting started. We're on panel number 4 this afternoon, and we're going to be speaking about perspectives on individualized therapies. The goal of this session is to provide various perspectives on individualized therapies with an emphasis on regulatory considerations. As with some of the other panels earlier today, I'll begin with some very brief introductions and then turn the floor over to our esteemed panelists to give a deeper introduction and some perspectives from them. And then we'll move on to questions. I have questions, but I also hope to hear questions both from you all here at White Oak as well as folks online. So briefly, I'll introduce myself. My name is Marika Kimbrell. I'm the Deputy Director of the Office of New Drug Policy, which was a recently established office in the Office of New Drugs. It came about as a result of our recent reorganization. And I'm serving as a panelist for the folks here. We've got a great group of people, and I think we're going to have a very good discussion. So we have Ella Basala, Petrulla Simboko, Julia Vitarello, Celia Witten, and Timothy Yu. We're going to introduce folks not quite in the order that we're sitting, because we discussed this earlier and it wasn't alphabetical, but we're sitting alphabetically. So first, Julia.
Hi, my name is Julia Vitarello. 3 years ago, my daughter Mila was given a death sentence. She was diagnosed with Batten disease, which is a rare neurodegenerative disease that's fatal and has no treatments, no cures. This was the same little girl who was a typical little Colorado girl who was hiking and skiing. She was swimming and biking and rock climbing by the time she was 2. She was advanced and normal and singing all the words to her favorite songs, to Puff the Magic Dragon. She was learning her ABCs. And at 6 years old, she lost her vision. And she was diagnosed with Batten disease. And I was told that she was going to lose very quickly her last words. She would say, "Mommy," for the last time. She would take her last steps, that we should buy a wheelchair, that her brain would atrophy and eventually be an empty skull, and that she would die in the next 5 years. As you can imagine, my life as I knew it was over that day. I was told that there were no tools in the toolbox. It was empty. There was nothing we could do. There was very little understood about this disease. So I started speaking with other rare disease parents who had been fighting for their children across many diseases. I spoke with scientists and doctors around the world. I read everything I could get my hands on around Batten disease, around lysosomal storage disease. And I learned pretty quickly that I needed to start a foundation and raise a lot of money. And I started doing that and telling Mila's story to anyone and everyone that would listen. At some point, I realized Mila was missing a mutation. She needed 2. It was autosomal recessive, and one of them was not able to be found. So in my desperate plea to find a lab that would help me find the missing mutation so I could test my 2-year-old son, who seemed completely normal at the time, and to be 100% sure that my daughter, in fact, did have Batten CLN7, a rare form of a rare disease, I reached out on social media and I crossed paths with Dr. Timothy Yu. And I will let him tell the story of, in a moment when he speaks about what happened after that. But in 1 year's time from when she was diagnosed, and I was told that no child had ever lived with Batten disease, 1 year later, we were moving from Colorado to Boston, and Mila was receiving a new drug tailored just to her called Milacin. And she became the first person in the world to receive a drug tailored to one patient. And suddenly, the no hope turned into a real second shot at life. Mila was 7 years old when she was diagnosed, when she was treated, and obviously had lost quite a lot. So she, is in a place where she's much better than where she should be right now. And I'm incredibly grateful, not only to Dr. Yu and his team, but to the FDA. I was told the FDA was going to slow progress, and instead, they absolutely played an instrumental role in making Mila's treatment happen and collaborated with Dr. Yu and his team. So thank you very much from myself, my daughter, my family, for giving her this second chance at life. And I just want to kind of end my thoughts here with, I stay up every night thinking about the millions of children like Mila. I don't know how it's possible that there are millions of children that are being diagnosed with rare diseases and dying every day. And I think about the possibility that a treatment like Mila's might be able to actually help some of those children, but some ends up being quite a big number when it adds up. And I see this as a really exciting time of opportunity and obligation to really explore a path of allowing treatments like Mila's to be able to be accessible across many, many rare diseases, and hope that there will one day be a tool in the toolbox for many of those families that otherwise would have no hope.
Thank you, Julie. I've heard you speak before, and I just wanted to say that every time it's almost more and more powerful. So thank you for joining us. So we've obviously heard from a parent and a caregiver and an exceptional advocate. And so now let's turn to the sponsor investigator perspective.
Thank you, Marika. And first of all, I want to thank all of you for the chance to come and participate in this Rare Disease Day here at the FDA. I'm up sitting on this stage as an example of a physician scientist who's been able to take advantage of new tools that are available to us in 2020, actually beginning in 2017, but in this new age, they're really wonderful tools for drug development that are available to us, built off of many people's decades of hard work. We are also in the privileged position of having wonderful diagnostic tools, being able to sequence genomes for patients and find the answers that were often so elusive in many people's diagnostic odysseys going up to this point. And thirdly, in addition to having the privilege of having great therapeutic tools and diagnostic tools, really benefiting from a time of renewed flexibility and innovation on the part of the FDA to think about creative ways to apply these tools. I want to fill in some of the details as to how we came to develop what folks are saying is one of the first examples of a truly individualized genomic medicine for Julia's daughter, for Mila. We were fortunate to meet her off of social media in January 2017. And we were fortunate to be able to offer whole genome sequencing for her that not only established a diagnosis, not only named the rare disease that she had, but also pinpointed the exact mutation that Mila had. It's an interesting time. It used to be that once we categorized the disease, that was essentially where you stopped. You would be able to provide a diagnosis, a prognosis, and you would begin working on, say, a gene therapy or a small molecule approach. And I think it's an interesting time because the reason I say that is that now being able to pinpoint the mutation, as in Mila's case, sometimes allows you opportunities that we didn't know existed previously. You've heard in earlier sessions how there are 7,000 different rare diseases, and it's wonderful that we're able to name them and diagnose them, but tackling them one at a time is a daunting task. Well, we were able to find in the instance of your daughter that she had an unusual type of mutation. It's a type of splicing mutation that afforded a mechanism to potential treatment that didn't require us to know that much about the disease process, to know that much about the mechanism of this defective gene. And it allowed us to develop a drug for her modeled after a drug that you've already heard about called Spinraza for spinal muscular atrophy, and customize it quickly for her based on studies of her own tissue samples. And with a lot of support from many people in academia, in industry, and from the regulatory space with the FDA, the Division of Gastroenterology and Inborn Errors, we have been treating her with this for the last few years. And to what we believe is meaningful impact on her course of disease and improvement in her quality of life. So I'm up here to share a little bit of this story with you from the standpoint of a physician investigator who sees this as a wonderful opportunity to to develop creative different ways of tackling rare disease. We've talked about finding the gene, about developing therapies, about understanding the natural history. Well, one other potential tool here is to develop treatments that might work across multiple different diseases that can be applied depending on the type of mutation that you might have. In Mila's case, a particular type of splice mutation. But our tools are increasingly being matched not just to the disease, but to the type of mutation. Single letter changes in the genome can be fixed in principle with CRISPR gene editing. Nonsense mutations have other approaches. Splicing mutations, like Mila had, can be addressed with yet other approaches. So I'd like to I'm using my time on the stage here to encourage the room, folks in the room, advocates, industry representatives, and the FDA to continue along this vein of flexibility and thinking about creative ways to apply these new opportunities.
Thank you, Dr. Yu. So now from an investigator to an actual patient. Ella, would you take the next turn?
Sure. I received an individualized therapy called Phage Therapy in January of last year. If you're not familiar, Phage Therapy is the use of a very specific virus to attack a specific bacterial host. I have cystic fibrosis, and this disease is characterized by bacterial infections in the lungs predominantly. And over time, these bacterial infections lead to lung damage, lung scarring, and then eventually respiratory failure. So over the past 2 to 3 years, I've been dealing with progressively more severe and more frequent lung infections. I had been using intravenous antibiotics to treat these infections and using longer courses of treatment and for obviously more stronger therapies as well. And I knew that this wasn't sustainable for the long term with antibiotic resistance. And so I heard of phage therapy in November of 2018. And I saw a documentary of a patient with CF who was treated by Yale University researchers. And I contacted these researchers, and I was very interested in having this therapy. And I communicated directly with them. They were very receptive and open to answering my questions about how this therapy might interact with my body and with the bacteria in my lungs. And I have a biology background, and so I think that was helpful and made me more confident in understanding this therapy and making the decision to receive the treatment. I didn't have the support of my doctor at VCU, my pulmonologist, my CF doctor, because he wasn't familiar with phage. He, didn't support, that there's no evidence to it, right? I mean, it's an experimental therapy. It's not FDA approved. But I decided to go through with the therapy. So by January of 2019, I was very ill. I had been dealing with a very resistant infection on multiple weeks, like over 5 weeks of IV antibiotics, and not seeing any relief in my symptoms. I was on supplemental oxygen 24/7. I was having fevers. I was doing breathing treatments every 2 hours to try to clear out the lungs from just filling up with mucus. So at that point, I knew the benefit would outweigh the risk of trying a treatment that really only a handful of patients had tried before me in modern history in the U.S. And so, it was kind of where I was in a dire situation, and that was certainly a factor in my decision to pursue it because I knew I needed an alternative. And so I traveled up to Yale University to receive treatment. And Yale had offered for my doctor to deliver the medicine or to give the medicine to me at at VCU at my home hospital, but because he was unwilling and unable really to navigate the EIND process and filing the appropriate paperwork, protocol process, and as far as also the IRB with the hospital at VCU, he was not able to do this for me. So I traveled up to Yale, and it was difficult because I wasn't sure if I was even going to be able to physically make the trip. I was that ill. But long story short, I received the therapy, and within about a week's time, I started clearing that particularly terrible infection that I had. And because of my positive outcomes, I've really been an advocate for bringing this therapy to a larger number of patients that are in need or that are facing, antibiotic-resistant infections and are dire need.
Thank you. So I think listening to you, I was reminded of a couple of things from this morning. One is clearly being a patient scientist yourself, and the other was Dr. Marks's earlier mention of this being a good example of what is often thought of as a more common condition, as once broken up becomes something rare, and in this case truly individualized. So thank you. So now let's turn to hear some perspectives of some esteemed American regulators. So why don't we start with Dr. Witten?
Yes, thank you. I'm Celia Witten. I'm the Deputy Director of the Center for Biologics at FDA. And as Dr. Mark said in the previous session, that's the center at FDA that regulates blood products, vaccines, and cell and gene therapy. So in particular, in the area of individualized therapies, some of the products that we have under our oversight would include phage therapy, gene editing, gene therapy, and some vaccines for treatment of cancer. And all of these therapies or potential therapies have an increasingly important potential role that they can play in the lives of patients. So we really need to try to pay attention to how we can facilitate their development and availability. And some of them, of the kind of therapies that I just mentioned, some of them may be made for a specific patient, and some may be customized for a patient. But either way, they're really made for an individual patient or a small group of patients. And a lot of these, the typical development path followed it's not the traditional drug development pathway, which is maybe development by a pharmaceutical company. But a lot of these products begin in an academic lab, and a lot of the activity is in an academic lab. And as I think we've just heard from the two patients or family members who've just spoken. And I don't know if the current system, we'd like to, work to try to facilitate availability for treatments for all patients who need it. The regulatory framework that we have right now, I think, has the flexibility to accommodate these kinds of treatments, as we just heard two examples of. And we have other examples in our center, things even that have gone through to licensure that maybe aren't quite individualized therapies, but have had some similar kinds of aspects. CAR T cells for certain kinds of cancers or cord blood would be examples where we've exercised considerable flexibility. So what is needed to facilitate development of these products and availability to patients? I think that's one of the things we're all interested in doing or hearing about. One thing, just make a couple observations. One is there's a lot of different models of collaboration right now, and some amazing individuals and organizations who are leading these collaborations to develop individualized therapeutics, and also a number of models for data sharing. I think we need to learn from these examples to find a way to develop these products as efficiently as possible. But I think it's possible that some new models of collaboration may be needed too, that include different stakeholders from who has traditionally been included. I mean, I'm struck, not just at this meeting, other meetings I've been to, by the really heroic roles that have been played by family members and patients advocating for their families and themselves. But I think we'd like to we'd like to think eventually that there could be a system where you don't have to go to these heroic efforts to have a treatment available. I mean, people shouldn't have a second job of finding treatment for their family members or themselves. So that's should be a goal we all, think about, although there's certainly room for many kinds of models. So one thing Dr. Marks mentioned in the previous session, and I'm just going to reiterate, that we're having a workshop, CBER is having a workshop on March 3rd to discuss some of these issues in further detail, both the technical issues related, and these all echo themes that you've heard so far about manufacturing. We hear estimates of manufacturing capacity for AAV vectors for all the projects that people would want to do at this time. We've heard from investigators that they have to wait in line for availability of the vectors. And I've heard anywhere from we need 10-fold more manufacturing capacity to 1,000-fold more manufacturing capacity just for AAV vectors, which is a promising area for gene therapy development. So the manufacturing issues, preclinical testing, clinical testing, and collaborations will all be discussed in this workshop. And I don't want to go on for too long because I know we have another speaker and we want to take questions. But I just do want to mention one thing that is important also to keep in mind, which is that we have really, when we think about these applications, we really think about that we should have two goals here. One is if there's an individual patient or a small number of patients who are in need, it's what does that patient or that small group of patients need right now, and how can we facilitate development of that product for that patient or that group. But a related question, and I think it's also important, is how can we leverage what we learn from that one patient or one program that will help us for the next patient and the next program? Because many of our products and our product applications share something in common. If you develop a gene therapy with an AAV vector for a disease or for a related disease with the same mode of delivery, hopefully you will be able to learn something from one application and you can apply to another. And there's a lot of challenges in doing this. I mean, phage therapy is a good example. How do you learn? Each patient may have a different resistant infection. There may be a treatment developed on an individual basis for them. But still, I think we want to figure out as we go through this and as we keep in mind the needs of each patient and their family members, how are we going to also keep in mind the next patient and the next patient after that?
Thank you, Celia. So now we'll turn from CBER back to CEDAR.
Hi, my name is Petrila Zboku. I'm a clinical team leader in the Division of GI and Born Errors products in the Office of New Drugs in CEDAR. Thank you very much for the invitation to speak here. So by way of background, I'm a pediatric clinical geneticist and I practice clinical genetics for several years, and also I was involved in research before joining the agency. And so I think from my perspective, the case of individualized or targeted therapies is very, dear and near to my heart, and of course, very, very fascinating. So I was fortunate enough to be involved along with, collaborate with team on the application for Millicent. This was a tremendous opportunity for both myself and the team to learn from both team and his group, from the family, from Mila. We all were very invested in trying to, figure out how can we best, fill the gaps and truly pave a new, a novel way of looking at this case. So obviously, this individualized or targeted therapies is a very, very novel approach. And I think, what you're hearing today is really a very good example of collaboration, but also creative thinking, and really being as flexible as one can be, keeping in mind the end goal and also the end result. Right. So I guess, first of all, I know the panel previously talked about natural history studies and delineating the disease. So In Batten disease, there are some natural history studies, and there's, of course, observation in day-to-day clinical practice from physicians. So in that case, and of course, Julia knows this better than anybody, there was kind of a clear path of where this would lead. And so the question, at least from our perspective and the team's perspective, is, what do we need at a minimum to make sure that we satisfy the regulatory requirements, the way that FDA interprets the regulatory requirements, and of course, place those in the context of this individualized therapy and the individualized and the individual patient. And so, in that spirit, I think from our clinical team, our toxicology team, our clinical pharmacology team, so everybody kind of came together, and we actually had very frequent interactions with Tim and his team. And it was one of those times that I feel like I was just so excited to be involved in this that, as soon as update came from Tim about, what's going on, how Mila's doing, and what dose should we do next, I would drop everything and just read it and, try to figure out and email people and say, okay, here's what we have now, what should we do next? And so, it was, great, of course, very satisfying because for myself, I've seen those children, I've diagnosed those children, and so I very well know, how the patients and the families think. So, from a regulatory perspective, it becomes really difficult. This is novel. We are not used to it. The FDA is not used to it, right? It's a novel paradigm. And the question becomes, do you fit this into your traditional paradigm, or do you create a brand new paradigm, right, for those individualized therapies? And the question is, you probably need a little bit of both, which is what our team also did. You really have to understand the regulations deeply to really know, why was it a regulatory requirement? It's not only, okay, well, you need 2 studies or in 2 different species for toxicology, but why is that? And what could we do, to get the minimum amount of, let's say, toxicology data, right? To have some assurance of safety so that the patient can get treatment. So I feel like our team really did a fantastic job really coming together and thinking through this, and not just saying, well, that's a requirement and that's the end of it, because that just wouldn't work, and we knew that. So a lot of lessons learned, I would say, from this case, and we continue to learn as we and of course Tim and Julia follow Mila and how she's doing. It's been very inspiring for all of us, and I'm sure everybody in the room would agree. And I know that from the part of CEDAR and OND, there's active work being done to bring people together to think through those challenges and also think creatively and realistically about how we can help, in this process to move those therapies forward.
Thank you. So listening to the 5 of you, I've got 2 questions that have come to mind. The first relates to, I think maybe each of you referenced the need for flexibility and responsiveness from a regulatory perspective in these areas. And what that brought to mind for me was considerations of benefit and risk when you have previously uncharted waters in a treatment. And so could each of you sort of reflect on how your experiences with individualized therapies have brought how you've considered benefit and risk and how it might be different from other areas or maybe similar, and from your various perspectives as patients, as parents, as regulators, and as physicians?
Well, when I received the therapy, as I mentioned, I was in a dire need where my life was at risk, and so I think that's an important consideration when deciding when is the appropriate time to do an individualized therapy, or if at all, and weighing that benefit and risk analysis. For me, for phage therapy personally, it's something. This is not it is a new therapy, but it's been around for a while and it had been tested in patients before, so I wasn't the first. So I think that, that made me more comfortable with trying something that has been researched, has been shown to prove to be effective. Whereas, with a case like Mila's, that would have been uncharted completely. And so I think that is a big factor in determining whether, or understanding the risks and the benefits.
So I guess I can go next. So that's a really important point, and I guess it goes back to assessing the benefits versus the risks, because the risks, of course, you cannot really assess, by itself at any point in time. So from a regulatory perspective, I think every decision or almost every decision that we make, and at least the way that I think about it, is truly a benefit risk assessment. So in the case of individualized targeted therapies, and in this particular case with Millicent, we're dealing with a neurodegenerative disease, very severe, with quite clear trajectory. Of course, with variable progression, but we have a good idea of kind of what to expect in general. So I would say that in order to determine how much safety data, for example, you need to assess and whether an IND is what we call safe to proceed, and also what dose to use, how to escalate the dose. That's all a benefit risk decision. And also, what data are emerging, how's the patient really responding to treatment, what toxicology information do we have from animals to maybe guide our safety monitoring to put in that benefit risk determination. And then, of course, how does the patient themselves report, or if they're not able to self-report, what does the family think about how they're doing, either objectively, subjectively. And in that way, you can put kind of the picture together. I do believe, though, that at the end of the day, it's a truly benefit risk decision from the patient's and the family standpoint. I don't know that anybody else can make that decision, truly. And that's something that at least, and for Millicent and I communicate that very clearly, these are, the requirements. This is what at the minimum we want to see. We'll be in close collaboration, and we were. And we had a very open communication. But in terms of truly making decisions about increasing the dose or decreasing the dose, or making changes to the frequency, or decisions about continuing or not continuing the treatment, I think in terms of providing all the information you have, it's really the family's ultimate decision to really weigh what does the risk mean to them. Because, of course, patients with rare diseases, we know that very well. The definition of risk is very different than someone who may have a more mild, a more common disease with a very different trajectory. And so we recognize that their risk threshold is very different. And so I don't know that you can ever really truly appreciate that unless you're a patient with a rare disease. And so from a regulatory perspective, we can have an idea of what maybe the benefit is. We can have an idea of what maybe some of the side effects have been. But putting this together to actually make a decision in the case of an individualized therapy is really difficult. And the patients and the families are truly the ones who can make that decision. So, but just to point out that I think the risk a lot of times, as you said, can be defined very differently by different people.
Thank you, Patricia. I feel like you said a lot of what's been on my mind. Leading up to Mila receiving Millicent, I have to be honest that the risk-benefit analysis was pretty straightforward and black and white to me. The way I faced it was the risk of treating Mila versus the risk of not treating Mila. It was very specific to one person, to my daughter, and what was going to happen to her if she wasn't treated was very black and white. She was going to lose all of her abilities in a few years, and she was going to die. And the risk of treating her was an unknown. And Dr. Yu and I had a very good communication on a daily basis practically, and I feel like I was educated as much as possible on what the possible risks were. And we really didn't know. There was no other patient in the world receiving Millicent, and there was actually almost no other patient in the world receiving a drug like hers. I have to be honest, I never spent more than 1 second questioning whether or not Mila should start Millicent or not. there was no other option. There was no other treatment. And Mila was losing her abilities rapidly, by the week, by the month. She was losing her, down to 2 words, then to 1 word, and down to no words in just a month. Taking 5 steps, 1 step, couldn't step at all by herself. And so for me, this was a great opportunity. I was afraid that, that Mila was not having any pain. And so I was afraid that maybe, who knows, maybe Mila would start having excruciating pain. And that was scary to think about. However, it absolutely did not influence my decision whatsoever, because the other option is that she was going to lose all her abilities and die. And so it was a pretty easy kind of risk-benefit analysis on my part. I would just say that communication was really important in terms of being able to do my very best as a non-scientist, non-physician, of understanding what Milasen entailed, and believing that there was some reason to have hope that Mila could have a stable disease, or potentially much less quickly, I guess, declining as she was going at that moment in time. And so it did offer real hope. This was not voodoo, kind of wild stuff that, was an unknown. It was a legitimate hope that Mila was given. So I weighed those two, and it was pretty black and white for me.
I would like to reframe the question slightly the way that we think about it. But let me say it's, obviously always case by case. I think the facts are different. But it's really a risk-benefit decision in the face of a certain amount of uncertainty, or an amount of uncertainty. And that amount of uncertainty may vary depending on the application. And so to say who makes the decision or how you make the decision, I think it depends on a lot of things, but the uncertainty has to be taken into account. And there are other considerations too. For example, if you look at the spectrum of the products that I've mentioned are addressed in our center, from phage therapy to gene editing, gene editing can't be withdrawn. It's not something where you can do a dosing study and then stop it or anything like that. So, for these different situations, and you may have a different understanding in different cases about how likely it is to be a benefit. Like if you have a lot of uncertainty about risk, but a fair amount of certainty about benefit, you might be willing in some circumstances to act on that. So I think it would be hard to answer generally, except to say that I think the uncertainty about the information you have supporting the application is also important.
So a little bit of background on how we thought about benefits and risks as we were considering whether to offer this therapy, whether we had done sufficient work, had we done sufficient work to justify our offering this hope to Mila and her family. So just to level set so folks know what was done for this drug, we decided that we wanted to go after a splicing defect, a defect in the way that Mila's gene was being assembled. And there are very simple but effective tools for looking and studying splicing if we wanted to, using patient-derived cells. So to be very concrete about it, we took a very small skin biopsy from Mila and then grew those in the laboratory and then studied her gene with and without the drug. And we're able to show that upon application of our drug, the splicing defect, the gene assembly defect, would reverse itself upon application of this new investigational drug. So that gave us some strong basis for thinking there could be a mechanistic improvement here. We took it a step further, and we also shared with the FDA data showing that would her cells actually become healthier when you give them this drug, and we showed that this disease was known to change the way that cells recycle proteins. And in fact, in the absence of this gene working properly, cells would fill up with products that were meant to be recycled, but what never actually made it to be recycled. And so they would build up and accumulate trash, so to speak. And what we found was that treatment of her cells with that drug was sufficient to allow recycling to happen again. And so they actually got healthier in front of our eyes. We could see this, and we could build up a scientific rationale. This was not a shot in the dark. This was something that looked scientifically plausible. All that being said, there were still many, many unknowns, and that standard that I just described, while it was good in this particular instance, that's not always sufficient for a rare drug application to go through. But in this case, as we discussed, we were up against a disease where the natural history was very clear, the risks were very, very clear, and we could talk about them with Julia and Mila's father and work through what exactly all of this meant to you all. And it's that very individual discussion. I think that's the interesting part of this. Now, if we take an agency which holds great institutional knowledge about proper drug design and safety at the level of populations, and now we throw in the application of that knowledge towards drugs that might just go to a single patient. The good news, I mean, the decisions are so personal and so individualized, and they have to do with the physician-patient relationship and how one talks through our assessments of these risks and benefits. But the good news is that the agency is populated with plenty of folks who have counseled patients on these exact issues in their own practices. And so I think that the novel piece here is figuring out how the regulations that apply to protecting public health apply in the situation now where you're juxtaposing that onto individual decisions by individual families. So I think that that's the part where, we're really grateful that you reached out on a limb to extend that paradigm to this case and looking for ways that we might continue doing that.
Great, thank you. I've got one more question, and then I think we'll open it up to the audience. But we're, as we predicted, eating up a lot of time, so maybe we can try to keep this one quick. But I do want to hit it. So one of Celia, I think it was you, talked about these situations often being successful when there's somebody heroic in the mix. Gluing everything together, or I tend to think of it as sort of the stars aligning in a particular situation. And I think our aim is that this shouldn't only be successful when the stars align perfectly, or when we have heroic family members or investigators or whoever working to make something happen. So what can we do, especially as regulators, and to sort of ease that process, to ensure that these treatments are available to the less than amazingly heroic among us, and then also what in that vein, what were your reflections on working with FDA and from the FDA, or sort of with the investigators, of what works well, what was successful, what comes to mind as important to keep in mind for an efficient process?
So I didn't have any direct communications with the FDA in my treatment. I really only communicated with the investigator. But I think, as far as my case, my doctor couldn't take on that role of being the investigator. And I think that this is quite common. It can be, I think, a daunting and arduous process to file the appropriate EIND, making sure your protocol is streamlined, and all the communications that come along with that with the FDA. I think a lot of providers or investigators aren't aware or aren't able to do this. And I think really, on the part of the FDA to help and create guidelines and really help them walk through this process so that more patients are able to access these treatments. I think, a lot of time, there is certainly roadblocks all along this. From, obviously I'm a patient that, really advocated for myself, but if I didn't have a provider that was willing to take on my case, or if the communications with the FDA weren't streamlined, it wouldn't have happened. So that's really like the stars had to align, as you were saying. And so, and also I think to remember that it's a resource intensive experience or process for the investigator too, because they have to communicate with the patient. I had to feel comfortable that he had my best interest in mind and my health in mind, and then he has to communicate with the FDA, and then along with the creation or the collaboration for the creation of the investigational therapies. So it's quite a bit on the investigator to navigate all that.
I was incredibly impressed with how Petrula and her team shifted from a paradigm that they were used to, where there was one drug for thousands of people, to an entirely new paradigm that they had never faced before, where the ripple effect was one. So it was Mila. No one else was taking Millicin. And they were able to change the way they faced the risk-benefit analysis and look at what the risk of treating Mila was versus the risk of not treating Mila, and really treat her as one individual patient, which reminded me a little bit of something like a brain tumor and removing a brain tumor. And, a doctor has a conversation with me that says, Mila has a brain tumor and we can take it out or we can leave it. And, having that kind of back and forth discussion of the risks of taking it out versus the risk of not taking it out, or for that specific patient. And I really applaud, Petrula and her team for shifting to a really different mentality that they were not used to, and really thinking about Mila. And as we move forward now, this has opened up a potentially new field of medicine, of really truly personal medicines. And what I see is I see Mila, and she was treated. And then I see potentially millions of children just with fatal diseases alone that could potentially, we don't know yet, could potentially benefit from a treatment like Mila's. And how do we get from Mila to really, truly making a difference, not just treating another 2 or 3 Milas, but really offering a tool in the toolbox across many, many diseases. And that's going to require working off of this new mentality and thinking, how do we face risk-benefit analysis when there is a 1 child or 2 or 3, and it's not being given to thousands of people. And so I just hope that, my hope is that we see more of this really out-of-the-box thinking, and really realize that most people like me don't have any options, and that this offers something exciting, but it needs very careful and very aggressive, pushing forward and opening up a new, potentially, field of medicine. So thank you for everything you've done. I hope it continues.
Okay. I thought I would like to say that cases like Ella's, cases like Mila's, these individualized cases, they require thinking in a way very small. They require thinking about individual patients' needs, about their particular assessments of risks and benefits, and they require thinking about treating that one patient in the doctor-patient type of way. But in a way, even though these individualized cases set a template and allow us to try something new, it's worth the walk only if we also figure out how to think big. And the question is, it's not that we want to convert drug therapists, drug developers' efforts towards now, instead of treating whole classes of disease at once, now just treating single patients at once. That's not the point of that. That doesn't make sense. Right? The point is that how can we leverage these wonderfully advancing tools to develop individualized treatments and then figure out how to scale it? And the true measure of success, I think, in this space, if we do this right, is that these first examples will pave the way for further examples, such that each example that follows it gets easier and easier, not more difficult and more difficult. I think that should be the goal of what we try to do from this point going forward, thinking about how, what are the lessons that we draw? Well, if you focus on one patient, you can get amazing things done. And can we use that to develop policies that allow that to scale, and so that each patient informs and makes the next patient's journey that much easier? I think that that's a really critical piece that I see coming from this.
Yeah, I mean, I wanted to add though, one important piece that I thought was critical in this is diagnosis, right? So Julia talked about how there was just one mutation found and you kept looking and you kept looking. And if you didn't do that, you wouldn't be here, right? So, and the diagnosis in many of the rare diseases and in the inborn errors of metabolism, which is the area that I work in, is sometimes actually quite challenging, even in this era of this, genetic revolution and of the tools that we have and the novel technologies. Because, even if you find one mutation or two, you still have to put it in a context of the patient themselves. What we see sometimes is that there's no specific guidelines or of how to diagnose some of those patients or how to treat them, how to follow them over time. And that really becomes an obstacle when you try to really look at a specific patient's mutation, for example, and kind of inferring how the patient may progress with their disease over time. So I think, what we're discussing today about Mila's case and other cases is actually a great example of all the fundamental concepts that I think we discussed throughout the day today, which basically is the diagnosis is really the most important step, and the awareness of the rare diseases, and really the outreach to patients who may not otherwise have access to some of those technologies through some infrastructure and networks, but also the role of really delineating the trajectory of disease. So if we didn't know anything about Batten disease, right, then that would be a very different story. And so the natural history of disease, we all talk about this, but it just becomes so critical, because if you don't know the trajectory of the disease and you don't know what to follow, when to follow it, what to focus on. And so, and the other piece of that is really the collaboration and the communication that I know we touched upon. And so, at least from, my perspective in our division, we're involved very much in outreach and engagement. And I myself, try to attend meetings and really learn from the outside. And so I think, at least for me, that becomes important because, you realize, what is it that the community wants? But what is it also the gaps that there are there, and how can we, as FDA, maybe help in that way to fill those gaps or bridge those gaps in some way? For example, I go to meetings and then I hear, some investigators talking about, well, FDA doesn't know what, we do and what we want. And, I'm kind of a fly in the wall and try to listen and say, okay, what is it that, we're just really not getting across? And some of those actually end up in guidances. And we wrote 2 guidances recently on inborn errors. And this is where a lot of kind of the engagement comes in to try to tackle some of those. And this actually trickles down to, how to bring people together, really, to work together in this very novel space. And the trajectory of this space is going to be, changing a lot. Of course, the basic principles would apply always. And I think we all need to be aware of what those are, but also how to apply them in a flexible way, in a creative way, and really a way that makes sense to kind of all parties involved.
So one thing I'll just mention that I know this comes up a lot in different contexts, but it's critically important here. And that's about sharing information so we can learn from what we've done. And it seems like it's always a hard target to hit, but in this case, it's really important because we can talk about learning from our experience or people learning from their experience working with FDA or us learning, how to work with people to develop these. But if we really want to, and I agree with what you said, if we want to scale this, then I think we have to think simultaneously about how we're going to learn from each of these experiences, not just about the process, but about, what we saw from whatever preclinical testing that was done or bench testing, and how that, what we learned from the patients.
I think that takes us back to Dr. Yu's comment of focusing on both thinking small and thinking very big at the same time. So on that note, if there's any questions from the audience, we'd be happy to take them.
Hello, my name is Amy Dom. I'm with the Cushing Support and Research Foundation. I myself am a Cushing's patient, and I also had postoperative adrenal insufficiency. And Dr. Stratakis at the NIH has been doing research that's showing that there are some genetic mutations that can at least contribute to Cushing's. So, given that, and given CRISPR, my colleagues and I have been wondering, what would that look like? Like, what would it look like if you took CRISPR and Cushing's, and would it be a complete prevention of it? Would it be a complete reversal of it? Like, would you have it one day and then the next you wouldn't? Like, what does it look like?
I think that it's, I'm optimistic that in the long run, we'll be able to figure out how to answer questions like that. But I think people are just starting to use, they have been under study for a couple of years, but we're just starting to learn about how to make gene editing tools useful. But the basic idea is to make a correction of the genetic defect in the cell, in each cell. The challenge, well, there are many challenges, but one of many challenges is how to get it to the cell, how to, if you can figure out how to correct the defect, how do you actually deliver it? So if you could perfectly deliver it and make these changes, then you would be good to go. But that there's a big different distance between where we are now and getting there. So I'm certainly optimistic that we'll get there, but that's the current state of things.
Thank you.
Do we have time for one more question? One more question.
Hi, my name is Christy Demarco. I was diagnosed with a rare, ultra rare disease. It's a 1 in a million back last year, and very quickly found out there was no research, no patient registry, no foundation supporting the disease. We started a patient registry, very much like my world changed at that moment. To try to create something that would actually, look for a cure for this disease. One thing I found, though, is it's interesting. As soon as we got the momentum going, that people are very interested in getting engaged. And, we've talked about a proof of concept for liver gene therapy, but a lot of it comes down to funding, And so I'd be interested to know from a individualized therapy. This is an ultra rare. It's like 1 in a million. So we found maybe about 20 some odd patients around the world so far. But being able to get someone interested in funding something like if we got positive results from a liver gene therapy, you're talking about individualized therapies. It's got to be a significant amount of money.
Yeah, I think that is a challenging area, which is why I think we have to discuss that collectively, because I don't, a lot of these, it's hard to see what the commercial model, what the model is for making these commercially viable efforts. So I don't think it's going to follow any kind of traditional pharmaceutical company supports it kind of model. But I know you have probably, Dr. You would have something to say.
Thank you for raising that question. It's one of the biggest challenges in the space. I think that what we have are issues of drug design, drug testing, manufacturing, toxicology, and then administration. And we have standards that have evolved for how to do those things safely and effectively. By and large, many of those standards, all of those standards have evolved with the best interests of patients in mind, to protect their safety and to ensure the utility of the drugs that actually make it through the pipeline. But I might argue personally that many of those standards have evolved in the era of big drugs like statins that might be given to tens of thousands of patients at a time. Now, I know that's an oversimplification and that there are many forward-thinking ways about how to apply these standards to smaller and smaller populations where that kind of investment can't be easily raised. But now take that drug development process and now apply it to a single patient for a family that's in dire need, and you've gone and blown up the problem even bigger. These are really expensive projects, and I'd say there's no norm. I mean, I'd say our first case of a brand new drug developed for a patient doesn't establish a norm. It just highlights an issue that needs to be solved, which is that it takes too much money to navigate all of those steps that I described. And we just have to be creative about finding solutions. I'll put in one brief plug. I'll say that for the particular route that we chose to use an antisense oligonucleotide drug approach. And that's an approach that has been around for about 30 years. The basic manufacturing process it relies on chemistry that was developed 30 years ago, or actually a lot more than 30 years ago. And as far as manufacturing processes go, I think arguably it's among the much simpler than, say, gene therapies. So you can mitigate the costs of manufacturing in a case like that, but on animal testing and toxicology, that's still extremely expensive. So finding ways to really make this work will require thinking about ways where we can leverage results from one experiment to another experiment involving a closely related drug, and another experiment involving another closely related drug. If we're learning from each of those and the learnings bolster one another, we should be able to shrink that gap. I don't know if it'll be sufficient, but we have to try.
Great, thank you. And I think we're about to be dragged off the stage. So I think Dr. Maynard's going to introduce the next panel. But thank you, everyone, and thanks to the panelists.